Hi, my name is Maroun Moussallem, and I will tell you today about some of the ongoing work at Dr. Shlenov's group at Florida State University. I will talk about how we were able to use polyelectrolyte multilayers to control protein absorption and cellular adhesion and motility. The protein we are using as a model is fibronectin, and the cells we are using are A7R5 cells. These are aortic smooth muscle cells extracted from embryonic rats. Polyelectrolytes are charged polymers, and polyelectrolyte complexes have been known for a long time. The idea here is if you mix two water-soluble polymers of opposite charges, like polydiallyl dimethyl ammonium chloride, which is a polycation, with polystyrene sulfonate, which is a polyanion, you will get an insoluble complex. But in the early 90s, Dasher introduced the layer-by-layer technique, which consists of sequentially depositing the oppositely charged polymers onto a charged substrate. This technique transformed polyelectrolyte complexes into polyelectrolyte multilayers and gave us control over a big number of properties. Some of the properties we can control are the thickness, the surface roughness, the chemical composition of the material, hydrophobicity and hydrophilicity, and many others. For this specific work, we are using different types of polyelectrolytes, ranging from very hydrophilic, like the zwitterionic copolymer, PAA copy-ADAPs, to very hydrophobic, like perfluoropolyvinyl pyridine and Nafion. We also use polyallylamine hydrochloride and polyacrylic acid. In order to quantitatively study the amount of fibronectin absorbing onto the different polyelectrolytes, we used a method called optical waveguide light mode spectroscopy. This instrument measures the encoupling angle at which the laser light encouples into the waveguiding film and gives a maximum intensity at the photodetector. From this angle, we can calculate the effective refractive index of the four-layer system. We already know the refractive indices of the waveguiding film, the glass substrate, and the covering solution. We also know the thickness of the waveguiding film, so we can calculate the thickness and the refractive index of the adsorbed layer, in our case, the proteins. In a typical waveguide experiment, I'm showing here typical waveguide experiment results of adsorption of fibronectin on the different polyelectrolytes. What we notice is that fibronectin absorbs much more on positively charged surfaces than on negatively charged ones. We also notice that as the hydrophilicity of the film increases, the amount of fibronectin absorbing on the film decreases, and zwitterionic films were protein repellent. As you know, the cell membrane contains between 50 and 75% of zwitterionic phospholipids. So we were interested to look at how the zwitterionic content of the films affects the behavior of the A7R5 cells. So as you can see, going from the left to the right, the A7R5 cells start to losing attachment and start being motile as the percent of zwitterionic content in the film increases, until they reach a certain point where the surface becomes cell repellent. Taking advantage of this behavior, we made a surface that has the zwitterionic copolymer as its outermost layer, and on top of that surface, using a micro-patterned stamp, we stamped 20 micron strips of Nafion. If you look at panel B, you can see that fibronectin only absorbs on the Nafion strip. In panel C, you can clearly see that the cells only attach on the Nafion strip. The zwitterionic surface was both protein and cell repellent. Using the same technique, we stamped the American Chemical Society and the Florida State University initials, and as you can see that the cells are very selective to the surface they grow on. In conclusion, we showed that polyelectrolyte multilayers can be engineered and fine-tuned to be used in different biomaterial applications. At the end, I would like to acknowledge all the funding agencies that funded this project, and thank you.